Everything's okay in Zoom? Okay. So today it's my pleasure to introduce Dr. Stefani Spranger. She did her doctorate at Ludwig Maximilians University in Munich, where she's from. Or not her doctorate, her PhD, yep. And then she did her postdoc with Tom Geske at the University of Chicago. Her focus has always been tumor immunology, human tumor immunology, mouse immunology, really pushing the boundaries of how we can understand cancer cell genetic events and tie that into T cells and myeloid cells. She started her independent research program at MIT in 2017. So we live in kind of parallel universes. And I'm excited to have Dr. Spranger here today, and I look forward to your talk.
Thank you, Ingrid, for this really nice introduction, and it's good to be back in the Midwest and the cold weather, honestly. I think I'm probably one of the few people that look forward to cold Minnesota weather. And as you said, I'm going to talk about what my lab's been doing. I'm just find my mouse to click. Okay, there we go. And today, the theme of the talk, hopefully that will come together, will be that not all T cell responses are equal, and that dendritic cells really matter quite substantially in this equation and how we shape the quality of a T cell response. But to refine this title a little bit, I want to revise the title to say tissue site really matters. And this applies mostly to anatomic location and where the lymph node is located throughout the body. So just to get everyone on the same playing field, I want to introduce the cancer immunity cycle and how we think about it from a very dendritic cell heavy perspective. So when we have a tumor growing in a human or an animal, the first thing that needs to happen to get an immune response activated is that dendritic cells need to sense the tumor, need to sense danger and infiltrate the tumor and pick up tumor cell debris. Upon activation of those dendritic cells, these then acquire a migratory program, which allows them to migrate to the closest draining lymph node, the so-called tumor draining lymph node. There in the lymph node, they see and present antigen, and CD8 T cells can recognize this antigen. This recognition then leads to a robust activation and proliferation of those CD8 T cells. They typically, and this is the canonical thought, acquire an effector phenotype, which then allows them to home back to inflammatory sites. And in the case of the tumor, this happens based on the chemokine gradient CXCL9 and CXCL10. And the chemokine receptor CXCR3 on the effector CD8 T cells. And then they home back into the tumor microenvironment where they can then exert effector function and lyse specifically tumor cells in an antigen-specific manner. Now, if this would work perfectly as I just described, cancer would not be a problem. We would not have cancer patients in the clinic. So often this cancer immunity cycle fails. And the most common failure that we think about clinically, and the one that we can actually target specifically, is that these effector T cells become exhausted or dysfunctional over time. So the acquisition of a dysfunctional phenotype in the tumor microenvironment is something that particularly immuno-oncologists in the clinic think about quite frequently. And this is because at this stage of the cancer immunity cycle, we actually have currently therapeutics that are very potent and very active to reverse this immune response. And these are so-called immune checkpoint blockade agents, namely PD-1, anti-PD-1, PD-L1 antibodies or anti-CTLA-4 antibodies. These block immune inhibitory receptors expressed on exhausted T cells, and then reinvigorate this exhausted T cell response to exert effector function again. This works really well in a subset of patients. And I'm showing here the poster child of immunotherapy, which is metastatic malignant melanoma. And as you can see here, this is historical data drawn in how chemotherapy looked in this cancer type. The first immunotherapy that was approved was just an anti-CTLA-4 antibody already rose this tail of the patients quite substantially up. So we have a lot of long-term responders to this therapy, and this keeps on increasing. If we now look at the neoadjuvant setting, For malignant melanoma, this actually goes up to like plus 70 or 80% of patients that are actually responding. So clearly melanoma is a fairly immunosensitive phenotype. When I started my lab, however, we were looking for cancer types where this is not quite as clear cut. And we got fascinated by non-small cell lung cancer. And we got fascinated by this cancer mostly because we see a fraction of patients that actually exhibits this durable response towards immunotherapy. But it's really only roughly 30 to 40% of patients. So it's not the entirety of non-small cell lung cancer. And we use this observation, this clinical observation, to generate a comparative model system where we can have patients that fail to respond and patients that do respond to understand why do the other patients fail to respond. And I want to take even a step further back and be a little bit more cancer type agnostic. And when we did that, and this is a lot of credit to one of my students who did all the work and keeps updating this chart actually based on clinical trial data, because this is pulled from many different clinical trials, and it summarizes the objective response rate across multiple cancer types to any type of immune checkpoint blockade therapy. And what you see here, the two cancer types that respond the highest to immune checkpoint blockade therapy are skin cancers, versus all the other cancers are still cancers of barrier tissue. So they have interactions with the outside world, but they are derived mostly in internal organ sites. And I want to draw your attention very specifically to skin melanoma, cutaneous melanoma, and non-small cell lung cancer. That's the comparison I introduced to you just now. And of course, there's many hypotheses on why two different cancer types respond differently to the immunotherapy. Probably the most common that you would hear when you talk to cancer biologists is the neoantigen burden is different between these cancer types, right? This is what we typically think makes a tumor immunogenic or non-immunogenic, and most correlative, this is related to non-synonymous mutations. We actually went into the TCGA dataset and analyzed the non-synonymous mutation burden between these two cancer types. And unfortunately, non-small cell lung cancer is separated in squamous and adeno in the TCGA. So you will have both of these entities here. But when you compare the amount of non-synonymous mutations between cutaneous melanoma and non-small cell lung cancer, they're roughly comparable. It's not that there's an order of magnitude difference between these two cancer entities. So while this still can be a factor on a per patient level, it's not a factor on the population level. The other factor that certainly contributes to the responsiveness of cancer patients to immunotherapy is the amount of immune infiltration, particularly CD8 T cell infiltration into those tumors. We have pretty good predictive signatures that can inform us whether there's a CD8 T cell infiltrate in the tumor. So we use the same TCGA data set to then predict, are those tumors actually CD8 T cell rich, or are they cold CD8 T cell tumors? When we overlay this, we actually see that, and the percentages down here are a little bit more clear, that non-small cell lung cancer has a higher amount of predicted CD8 T cell infiltration compared to melanoma, which is exactly the opposite of what we observe with a response to immunotherapy, indicating again that it's neither the antigen burden on a population level, nor the inability for CD8 T cells to home into the tumor microenvironment. So it must be something else about the tumor or about the tissue that actually restrains this immune response. And with all that pre-knowledge, I now want to dive deeper into what do we understand about the clinical immune microenvironment of non-small cell lung cancer. And I'm doing so with this pictogram, which shows you the range of the immune phenotypes that have been observed in non-small cell lung cancer patients. And there's basically 4 phenotypes, and I'm going to start with the 2 most clear-cut ones. This one here is the T cell infiltrated phenotype. This is characterized by the presence of CD8 T cells, as the name says, but also, and this is specific for non-small cell lung cancer, by the upregulation of PD-L1. And PD-L1 is a downstream interferon gamma responsive protein. So the more interferon gamma the CD8 T cells produce in the tumor microenvironment, the more PD-L1 we have as a negative feedback mechanism in the tumor. So this infers that these T cell responses are actually functional. And then the extreme other example is the no T cell infiltration, which has been observed in non-small cell lung cancer. It's mostly driven by LKB1 and STK11 mutations, and it's characterized by an absence of almost any adaptive immune cells, an absence of dendritic cells, and a high infiltration of neutrophils. Then we have this T cell excluded phenotype, which is a cool phenotype. We actually have a project on precisely that. And it seems that there's physical barriers by the tumor microenvironment put in place that don't allow T cells to actually enter into the tumor. But for today's talk, I want to draw your attention to this very last phenotype, which is a quite unique one. It has been described by Roy Herbst in the clinic in 2014, and it was originally described as a non-functional T cell response. And what that means clinically is there are CD8 T cells infiltrating the tumor. They seem to be clonally expanding, so responding to an antigen, but there's no PD-L1 upregulated in the tumor microenvironment. And that implies that these T cells don't exert effector function for one reason or another. But outside of that, we didn't understand how this phenotype is actually being established. And the majority of today's talk will focus on work produced by Brendan Horton, a postdoc in the lab, and Maria Zaruglia, a recently graduated grad student, who really pulled this phenotype apart and showed that this phenotype is quite related to a tolerance phenotype. So what Brendan did when he started the work was he wanted to specifically introduce and induce this immune phenotype in a mouse model of non-small cell lung cancer. So what we did is we used a low mutational burden cell line derived from an autochthonous lung cancer mouse model driven by KRAS, G12D, and P53 deletion. We then inoculated this tumor cell line either in the flank of mice or orthotopically in the lung by either intravenous injection, or we can also do intratracheal injection straight into the lungs. And then we treated the animals with dual checkpoint blockade immunotherapy as patients would be treated. And as you can see here, the lung tumor burden between control and treated animals does not change if the tumor grows in the lung, but it does significantly change when the tumor grows in the flank, which is intriguing because, again, this is a low mutational burden tumor cell line. It should not be very immunogenic. And then we looked at the CD8 T cell phenotype in the tumor microenvironment. And this is looking primarily first by number. What we observed is the lung tumor lesions were quite infiltrated at baseline, but immune checkpoint blockade therapy did not increase the amount of T cell infiltration significantly in those tumor lesions. In contrast, the flank tumors had fairly low T cell infiltration at baseline, but we saw a dramatic increase in the amount of T cells in the tumor microenvironment, indicating that in the flank setting, the T cells were susceptible for reinvigoration by immune checkpoint blockade. And we then wanted to understand what is different about those T cells. And we took the unbiased approach of single-cell RNA sequencing, and all the single-cell RNA sequencing you will see in this talk is done in close collaboration with Chris Love's group at MIT. And what we did is we single-cell sequenced all CD45 positive cells, but for this very specific question, I would like to draw your attention to this cluster of green and orange activated CD8 T cells. We then wanted to understand where do these two T cell clusters come from. Interestingly, we found that the orange cluster was primarily found in flank tumors, versus the green cluster really dominated the lung tumor microenvironment. And of course, you can now challenge this observation because we sorted out polyclonal CD8 T cells, that there's an antigenic difference. The tumor cells are not quite identical, although we start with the same starting population. So to address this, we engineered a model antigen into those tumor cell lines and then repeated the experiment, sorting out only the model antigen reactive T cells from the tumor microenvironment. And when we repeated that with the SIY model antigen, we observed exactly the same phenotypic distribution, indicating it's not the antigen that drives this response, but it's actually the microenvironment that drives this response. And we were interested in what are the orange versus green activated CD8 T cells. So we then did a differential gene expression analysis between these two clusters. And for the T cell biologists in the room, it's quite clear to see that the orange cluster resembled somewhere a T cell activation state along the trajectory of effector T cells or early exhausted T cells. Versus the green cluster over here, at the time, we're still somewhat confused, but we were quite confused in the beginning, but we think it resembled somewhere between a tissue-resident memory phenotype and a tolerogenic state. For the sake and the lack of a better term, we call this T cell dysfunction, because it's truly a dysfunctional phenotype when it comes to an anti-tumor immune response. We're actually able to validate those observations using flow cytometry. So here we looked at PD-1 as a sheer marker of T cell activation and antigen engagement. Both of those T cell populations do see antigen, but only the T cells in flank tumors actually upregulate canonical markers of T cell exhaustion, as well as markers of effector T cell function, while the cells that we find in lung tumor lesions have homing markers that allow them to go to the lung, which makes sense. And then we wanted to understand more, is this a function of the local tumor microenvironment? So are those T cells the same when they become activated, and then do they change in their phenotype once they're in the tumor? Or is this difference instructed into them earlier during T cell activation? And to do that, we again use the SIY model system, where we have TCR transgenic T cells that are specific for that model antigen. These T cells are called 2C T cells. We can label them with a proliferation dye and transfer them into tumor-bearing mice. And then 3 days later, we took out the tumor-draining lymph node and assessed A, are both activated, and B, how is the quality of this T cell activation? And you see here in this example flow plot, both lymph node locations, the inguinal lymph node for the flank and the mediastinal lymph node for the lung, activate T cells with roughly the same kinetics. But when we then did bulk RNA sequencing on those activated T cells derived from the lymph node, and I want to emphasize this is 72 hours after adoptive transfer, so fairly rapidly after T cell activation, we see very different transcriptional profiles induced. And again, when we look at the transcription factors and epigenetic modifiers here, as well as the cytokine receptors, the T cells activated in the inguinal lymph node resemble an early effector differentiation or early exhaustion differentiation, versus the blue ones activated in the mediastinal lymph node, again, have a little bit of mixed profile, having a lot of markers that derive them to the tissue resident fate, but also markers of longevity like TOX1 and TOX2. And we're still not quite making sense of the transcriptional changes that we see that early. And we can again use markers of T cell activation to differentiate these two states. Does this matter for immune checkpoint blockade therapy response? We wanted to address that question, of course, as well. So we treated both animal cohorts, the ones with lung tumors and the ones with flank tumors, with immune checkpoint blockade therapy, and then both in the tumor draining lymph node as well as the tumor assessed the activation profile of those T cells. And indeed, we found that particular granzyme is highly induced in the lymph node as well as the tumor if this is a flank tumor, but it's not induced if this is a lung tumor lesion. So truly, we have a developmental roadblock in the mediastinal lymph node that prevents these T cells from acquiring effector function, which is why we think they're somewhat related to tolerance. They're not quite tolerant because they're still actively proliferating and seeding the tissue. But does this matter for patients? That's, of course, a pretty high bar to prove because you can't just go and do adoptive transfer experiments in humans. But what we were able to do is we used a single cell RNA sequencing data set where in this case, this was 5 non-small cell lung cancer patients sequenced in the tumor microenvironment, and they found several different distinct clusters of CD8 T cells, and we generated a dysfunctional signature as well as an exhausted signature, and indeed found that these 3 clusters over here were highly enriched in the same transcriptional program that we found in the dysfunctional signature in the mouse model in those patients. So how can we therapeutically perturb the system? That was one of the questions we had to conclude this first observation. And one thing that struck us was that chemokine receptors are highly induced in the inguinal lymph node primed T cells. And these include CD25, as I showed you even on flow cytometry, but also IL-12 receptor chains. So we collaborated with a bioengineer, Dane Wittrup, in the building, who made half-life extended IL-2, IL-12 variants, and then gave those variants to lung cancer and tumor-bearing mice. And strikingly, only when we gave the combination of both cytokines did we rescue the phenotype, indicating that there's some synergy and some lack of the cytokine signaling locally in the tumor in the lymph node environment that prevents these cells from actually getting sufficient signals to push them over the bar to break tolerance and actually induce a productive T cell response. And strikingly, when we give this therapy, we can actually extend the survival of those animals. Again, this is a multinodal disease because we give systemic lung cancer cells into mice. So this is a hard model to actually cure. But what also is striking is we do not observe synergy with immune checkpoint blockade therapy, which might indicate that immune checkpoint blockade therapy and T cell exhaustion is on a different trajectory of T cell development than the observed dysfunctional phenotype that we have in the mediastinal lymph node. So just to summarize this initial observation that is the foundation for the rest of the talk, we found that in the inguinal lymph node combined with cutaneous or flank injected tumor cell lines, we get an effector CD8 T cell priming. These T cells acquire all the key molecules needed to actually eliminate cancer cells, and we get target cell destruction. And in contrast, when we get priming against the same antigens in the mediastinal lymph node, we get a dysfunctional T cell activation. They lack effector molecules as they differentiate and home into the tumor, and we do not get tumor cell destruction. And this is where my student Maria got really interested in how does this actually mechanistically occur in the tumor draining mediastinal lymph node? What happens there and what restrains the T cell activation? And the first thing we wanted to understand is it the same type of antigen presenting cell that actually mediates T cell priming? This is important because every tissue has its own and unique set of myeloid cells that seed the tissue, and theoretically there could be differences in which cell actually presents antigen. So we started with a simple experiment and had the hypothesis that cross-presenting dendritic cells driven by BATA3 cause T cell activation against tumor-derived antigens. And indeed, in both settings, here in the lung cancer setting as well as in the flank tumor setting, we saw that conventional type 1 cross-presenting dendritic cells were essential to prime CD8 T cells. So we were lucky in that regard. We were studying the same type of antigen-presenting cell. And then we wanted to understand what could be different about these dendritic cell, about those dendritic cells in the lymph nodes. So Maria engineered the tumor cells to express ZS green, which is a pH stable fluorophore, and allows us to track dendritic cells that come from the tumor, go to the tumor draining lymph node, and then present antigen to the T cells. And when she did that, we were first surprised that the mediastinal lymph nodes dendritic cells had more antigen. So that was one observation. They had more antigen. But then, more consistent with our phenotype, CD80 and CD86 was lower on the DC1s coming from the mediastinal lymph node. And so was IL-12. And that fit the bill. So our working hypothesis at that point was the dendritic cells coming from the lung, draining into the mediastinal lymph node, have an intrinsically different threshold to mount CD8 T cell responses. So what Maria then did is she established a quite heroic co-culture system where she sorted out tumor debris positive CDC1s from the mediastinal and inguinal lymph node and co-cultured them with TCR transgenic T cells. And I say heroic because whoever ever was trying to look for the mediastinal lymph node, it's tiny and you need a lot of mice to actually get sufficient amount of DCs. And what she found was, indeed, both dendritic cells are capable of priming CD8 T cells only when they have antigen engulfed, but the dendritic cells from the mediastinal lymph node were better at priming effector T cells than the ones from the inguinal lymph node. And that was exactly the opposite of result that we were observing in vivo. So we were quite puzzled by this. And then actually analyzed the dendritic cells a couple hours after isolation, and they recover CD80, CD86, and IL-12 quite rapidly. So we had to go back to the drawing board and think about a third party that restrains dendritic cells in vivo. And because we saw this strong observation of CD80 and 86, we thought, well, maybe regulatory T cells could directly interact with those CDC1s and restrain their stimulatory capacity. And we did the brute force approach first and used a mouse model where we can deplete all regulatory T cells by giving diphtheria toxin. And when we do this, all regulatory T cells are gone. Luckily, because of our adoptive transfer readout as a priming readout, we only have to do this for a fairly short period of time, so we're not actually inducing autoimmunity. But when we do this and then adoptively transfer tumor reactive T cells into those mice that are depleted for regulatory T cells, we can restore CD8 T cell effector priming quite substantially in the mediastinal lymph node. So then how can we prove that regulatory T cells are really essential for this equation? Because depleting all Tregs from a mouse, grantedly, is a sledgehammer and it's not physiological. So Maria went back to her co-culture system and now adapted the CD8 plus dendritic cell co-culture system by adding in the regulatory T cells sorted from the same draining lymph node. And when she did that, this here is the control setting where there's no regulatory T cells added. And you can see here, these mediastinal lymph node derived DC1s are pretty good activators of effector T cells. And when we add a 1:8 Treg to effector ratio, so a very low ratio if you think about Treg suppression, we restrain effector differentiation of CD8 T cells, but preserve proliferation of the CD8 T cells. And I'm sure many of you have used the conventional Treg suppression assay. How does this compare to the conventional assay? We actually did this comparison. So in teal is the CD3, CD28 activated, no dendritic cell Treg suppression assay. You can see that the first thing that goes away is proliferation, while we retain proliferation in our DC stimulated assay. While if we stimulate with CD3, CD28, CD25, and granzyme actually is pretty sustained, while we lose this quite rapidly. And this is one example where if we reduce a system down to the core backbones, we need to make sure what are we actually reducing it down to. So in this case, CD3, CD28 stimulation also activates the Tregs probably to superphysiological levels. So then we wanted to know, is this Treg suppression antigen specific? Because Tregs can simply be in the environment, soak up IL-2 and mediate unspecific suppression, or is this actually in an antigen specific manner? And to address this, the first thing we did is we used, again, this ex vivo presentation assay and used an antibody that blocks MHC class II. And when we did that, and the Tregs, this is all done in a V bottom plate, so they are in very close proximity to one another. When we do this and simply block MHC class II Treg interactions, we restore CD8 T cell priming, which was quite striking. We also validated these observations using a transwell assay that contact is really, truly important for mediating this phenotype. We then wanted to know, is contact also important for the in vivo observation. This was a little bit a harder bar to prove, but we achieved this by using a congenic marker on the adaptive, the transferred CD8 T cells. These are shown in white. In green, we have cross-presenting dendritic cells in the corresponding draining lymph nodes. And then in magenta, we have the nuclei of regulatory T cells by staining FOXP3. And what we did is we identified clusters of proliferating CD8 T cells, then identified which is the closest fragment of a dendritic cell, because you never catch an entire dendritic cell in a lymph node. That's sheer impossible. And then generated a radius around this dendritic cell, and then enumerated the Tregs and measured proximity between the Tregs and the cross-presenting dendritic cells. And indeed, we found that in the mediastinal lymph node, regulatory T cells are closer to priming dendritic cells than they are in the inguinal lymph node. And it is also only when those dendritic cells are in close contact with proliferating CD8 T cells, indicating that there is some micro-niche formation in the lymph node where CD4 regulatory T cells come together with CD8 T cells on the same dendritic cell, and that interaction determines the quality of the CD8 T cell response. So we then wanted to know, can we prove this in vivo a little bit more? Because microscopy is nice, but it's not definitive proof that this actually matters. And the first thing how we approached this was we made mixed bone marrow chimeras, mixing wild type and MHC class II deficient bone marrow together. These were on two different congenic markers, so we were able within the same mouse to compare the quality of the DCs that is able to interact or not with regulatory T cells. And when we did this, we phenocopied the phenotype that we observed between the inguinal and the mediastinal lymph node, meaning the dendritic cells that were capable of interacting with regulatory T cells had lower CD80, CD86, and IL-12 compared to the dendritic cells that were unable to interact with regulatory T cells. But does this matter for CD8 T cell priming? And this is, I promise, the most complicated bone marrow chimera approach that you will see. And really the important group is this one here to focus on, where we mixed MHC class II deficient bone marrow with bone marrow from a mouse that lacks the transcription factor BATF3, so it does not have cross-presenting dendritic cells. And once fully reconstituted, this immune system has MHC class II expression on all antigen-presenting cells, except for cross-presenting dendritic cells. But it still has cross-presenting dendritic cells coming from the MHC class II negative bone marrow, allowing for CD8 T cell activation. So when we use these animals and then transferred in tumor-reactive TCR transgenic T cells, we now restored CD8 T cell priming, indicating really that regulatory T cells on the same dendritic cell restrain the stimulatory capacity of that particular dendritic cell, and by doing so retain CD8 T cell effector function. But now I hopefully convinced you that this interaction is real, but what I have not showed you any evidence for is why regulatory T cells in the mediastinal lymph node are actually different compared to the inguinal lymph node. Because, right, regulatory T cells are throughout our body, they're throughout all lymph nodes. What makes them different in the mediastinal lymph node? So for this, we again collaborated with Chris Love and studied the regulatory T cells in a much more granular fashion. The first thing that we were interested in was, are the numbers different? And short answer, no. We didn't see any differences by number nor by percent between the two lymph node microenvironments. And then we used, again, single cell sequencing combined with TCR sequencing, and this was actually quite essential for finding the right population of Tregs, to understand what population of Tregs is responding to the tumor. Just to walk you through here, we sequenced, and this is all from lymph nodes, we sequenced tumor draining lymph nodes in the darker colors and naive lymph nodes in the lighter colors. We found 4 different clusters of regulatory T cells that are activated, these 2 being more early activation and proliferation, and these being truly activated. And then by using TCR clonotype analysis, we identified that upon tumor challenge, only this bottom cluster here is actually clonally expanding. It's expanding in both lymph nodes. So we were quite surprised that we actually see even public clonotypes between the 2 models. But it indicated that this activated cluster 1 is the one cluster we actually need to focus on. And then I'm sparing you the volcano plots, but we did differential expression analysis of the yellow cluster, of the C1 cluster, between the 2 lymph node environments, and identified that there's a skewing towards a Th1 effector Treg phenotype in the mediastinal lymph node. And to convince you that this is really true, I'm going to show you the flow cytometry validation, where we looked for T helper transcription factors, TBET, GATA3 and ROR-gamma T. And you can see that only in the mediastinal lymph node, we see the skewing of higher TBET levels in the regulatory T cells. And this higher TBET also goes along with a higher expression of CXCR3. And we believe the chemokine receptor expression levels that are associated with the Th1 phenotype are actually essential for driving this phenotype, because again, we observe this proximity observation that they need to come together at the same dendritic cell, which probably makes the same chemokines attracting both. The Th1 phenotype is downstream of interferon signaling. So we wanted to understand, is this interferon gamma or interferon, type 1 interferon? We again did a comparative bone marrow chimera approach, where we compared wild type to interferon alpha, gamma, or double knockouts, and found that the main driver of this response was interferon gamma sensing on the regulatory T cells. And then wanted to understand, are the levels of interferon gamma between the 2 lymph nodes actually different? And here we did an experiment where Maria harvests quite rapidly the lymph nodes from tumor-bearing mice, puts them through a cell strainer, and then spins them down quite rapidly, and then collects the fluid that we harvest and simply measures interferon gamma by ELISA. And when we do this, we find more interstitial lymph node derived interferon gamma present in the mediastinal lymph node compared to the inguinal lymph node. We're like, so there's some innate lymphocyte responding to the cancer insult, and that could potentially explain our entire observation that the mediastinal lymph node has more Th1 Tregs. So we went and looked in a naive lymph node where we expected this not to be present, and we're surprised that we still see the same differential, in suggesting that this is a steady state observation where we have interferon gamma produced by some cell, either in the lung microenvironment or in the mediastinal lymph node directly, that produces interferon gamma. And this is not the case in the inguinal lymph node. And you can see that we specified here that this was done in SPF, so special pathogen-free animals, because when we now do the very same experiment in naive germ-free animals, this difference is entirely gone, and the mediastinal lymph node looks like the inguinal lymph node. And this suggested to us that there's some commensal microbe that is being sensed by the immune system in the lung microenvironment at steady state that sets a certain threshold of tolerance for the lung microenvironment. Maria then also, of course, wanted to see if we can somehow use this therapeutically. And this is still far away from optimized, but it is a proof of concept that we can theoretically target this suppression mechanism therapeutically. So what she did is she used an interferon gamma blocking antibody very early after tumor inoculation to change the Treg phenotype. And you can see here, it doesn't change the number of regulatory T cells, but it reduces the TBET expression and the Th1 skewing of the regulatory T cells. And when she then transfers in tumor-reactive T cells, these acquire a more effector-like phenotype. And one of our future directions is actually to go back and see if there's any cell surface markers on Th1 effector Tregs that we can more specifically target to get rid of only that Treg population, but not the entirety of regulatory T cells. Again, this is also important for humans. This is actually a melanoma data set. Where we see a similar trend observing, where we look at just the CD8 to Treg ratio, which is a very common clinical biomarker that was unable to predict non-responders from responders. However, when we look at the quality of the regulatory T cells, either if they have an interferon gamma sensing gene signature or just simply TBET in the regulatory T cell compartment, we can predict whether these patients are non-responders or responders, indicating that the same is true for regulatory T cells. The quality of the regulatory T cell response probably matters more than the quantity of the regulatory T cell response. So with that, I would like to summarize this part of the talk, where we were able to show that in the mediastinal lymph node at baseline, we have higher abundance of interferon gamma. And I want to point out this interferon gamma is far lower than a full effector CD8 T cell derived interferon gamma signature, which is low tonic interferon gamma, which skews the regulatory T cell compartment into a pre-activated Th1 effector-like phenotype. And these effector Tregs are better or faster at suppressing CD8 T cell priming and probably help to sustain tolerance in a tissue-specific microenvironment. And this response is absent in the inguinal lymph node, so we get a more productive CD8 T cell priming. And with that, I would like to pivot to an unpublished story that we're currently writing up, focusing on the same model system, but now specifically looking at the flank tumor response. So a much more artificial tumor response, but a very unique model system, because we get very drastic CD8 T cell expansion, which allows us to study large amounts of tumor reactive T cells in a systemic fashion. And just to preset the story a little bit, this is how we currently think about T cell exhaustion and cancer immunity in a much more refined manner. So we believe that a naive T cell gets primed in the tumor draining lymph node. It there differentiates into a precursor T cell phenotype, which has stem cell capacity and can give rise to all T cell fates downstream. Then it commits to a progenitor exhausted T cell state that is already committed to enter the T cell exhaustion fate. And these progenitor T cells are still believed to be in the tumor draining lymph node and are currently the main target, or believed to be the main target, for immune checkpoint blockade reinvigoration. So basically pushing checkpoint blockade immunotherapy pushes differentiation of those progenitors into more effector terminal exhausted T cells. And then this is from an actual review. The lymph node drains straight into the tumor, which is anatomically absolutely not correct. And then in the tumor, you get full differentiation in the environment of high antigen abundance with a lack of co-stimulation. That's the current thought of the field. And while we were looking and accumulating data for the first two stories that I showed you, we made one observation that made us quite curious, because we take the spleen often as a control organ. But what we found is that when we compared the amount of CD8 T cells, the number of CD8 T cells that are tumor reactive in the lymph node between the two model systems I showed you, there's no difference in number. If we look in the tumor, there's no difference in number. If we look in the spleen, there's a significant difference in number. So we started to wonder, is there something about the spleen or the circulation that allows T cells to change their trajectory and fate? And is there something about this anatomical location that is important for how exhausted T cells develop? So this is now, from this point on, all the flank system, because again, we get large numbers of conventionally exhausted T cells. When we treat with immune checkpoint blockade immunotherapy, just as a reminder, we see a reduction in tumor burden. But then when we look and analyze the CD8 T cell response, depending on anatomical location, we see the largest delta in number of T cell changes following immunotherapy in the spleen. This is a over 10-fold increase in the number of T cells. So we became even more curious about this organ site. And again, this is just to summarize what 2 recent papers have put forth on how the exhausted T cell trajectory occurs. And we actually, to preset this, agrees with this trajectory. There's a precursor, there's a progenitor, there's 2 intermediate phenotypes. And then likely out of one of those intermediate phenotypes, we get terminal differentiation into terminal exhausted T cells. Both of these studies predominantly used T cells from chronic viral infections. And here the spleen, and this is why I think we're contributing quite substantially to the tumor field, the spleen is a major site of inflammation and viral production. So it's not the same anatomical location as it is for a tumor response. So we used, again, single-cell RNA sequencing combined with TCR sequencing to probe this. And this here is just showing the different clusters and that we actually see the same T cell clusters appear in our data set. And then we first binned them back together and looked, where do we see expansion? So this is here the pre-exhausted or progenitor or precursor T cells. We see them primarily in the lymph node, consistent with the literature. We see the progenitor T cells also mostly in the lymph node. The intermediate state, and this is where it becomes interesting, is mostly localized in the spleen. And then the terminal exhausted T cells are, as predicted by all models and all published data, mostly localized into the tumor. So then we refined, because we were really interested in the intermediate and exhausted phenotypes, we refined our analysis and identified that upon checkpoint blockade immunotherapy, it is the intermediate 1 subset that actually changes the most, and it changes the most dramatically again in the spleen. The intermediate 2 does not really change all that much between the treatments. Exhausted 1 also does not change all that much, and also quite interestingly, the KLR exhausted, which has been reported also to be a phenotype in chronic viral infection, did not localize to the tumor, but it did localize to the spleen in our hands. So we then wanted to use cell surface markers to study this much more dynamically in mice. And this is also consistent with the 2 stories, the two papers that I showed you earlier. We can use 2 chemokine receptors, CXCR3 and CX3CR1, to differentiate the intermediate 1 progenitor and exhausted KLR exhausted phenotypes in the spleen. And as you can see here, we mostly see CXCR3 single positive progenitor-like T cells in the lymph node. We still see them in the spleen to some level, but here they really upregulate the CX3CR1 molecule, entering into the intermediate phenotype in the spleen, and then in the tumor, they're becoming true double negative terminally exhausted T cells. So we then use the TCR clonotypes sort of as a barcode to identify clones that are in between the transitions to allow us to then piece these puzzle pieces together in a more systematic manner. And we identified that the intermediate 1 to exhausted 1 transition is the one, it's literally the only one that changes upon immune checkpoint blockade immunotherapy. And this is just to show this in a different manner, but where are those cells? Because, right, this is how I started. We made this spleen observation. And we find when we look at the cells that transition from intermediate 1 to exhausted 1, we find these cells primarily in the spleen and the exhausted ones primarily in the tumor. And just to show you this is not a general phenotype, when we look at the clonotypes that are between intermediate 1 and exhausted KLR, we find the intermediate still in the spleen, but then they're retained in the spleen when they become KLR exhausted. And then we wanted to know, what is it about the spleen microenvironment? So we first set up, again, a model system where we can use adaptive T-cell transfer to piece this apart. And this here is just to show you that the adaptively transferred T-cells phenocopy the endogenous T-cells. And then we can use a serial transfer approach to probe, is the spleen a unique reservoir of T-cells that is very potent to give rise to productive anti-tumor immune responses. So what we did is we activated the T-cells, transferred them into secondary hosts, and we did this by separating them coming from the tumor-draining lymph node or the spleen, and then treated with immune checkpoint blockade immunotherapy. And when we did that, and this is a quite surprising result, is only when we transfer T-cells coming from the spleen, so likely of this intermediate 1 phenotype, were they able to seed all tissues and productively expand upon immune checkpoint blockade immunotherapy. We still don't quite understand what's so unique about the splenic microenvironment. One hypothesis that we had was it's low in antigen, and this might still be true. It's not absolutely negative. We see some antigen-presenting cells that carry antigen. But we wanted to know what would happen if we blast the system with much more antigen. What would happen? Because now we're modeling the chronic viral response. So we basically just pulsed splenocytes and gave them through adaptive transfer. So now in this case, splenocytes are our antigen carrier. And we basically induced or shifted the transition from intermediate 1 to terminal exhausted, able to go to the tumor, to KLR exhausted. As you can see here now, when we give antigen, we see a reduction in CXCR3 and an increase in CX3CR1, indicating that they're now in this KLR exhausted phenotype. There's indication that this KLR exhausted phenotype is a non-productive immune response for cancer because you lack the ability to actually home into the tumor microenvironment. And probably for these types of immune responses, we need a different set of immunotherapy to actually get those T cells back into the tumor microenvironment. So with that, I'm leaving you guys with this working model where we think we corroborated the exhausted T cell trajectory that has previously been reported in viral infections. In a cancer model, we know that progenitor cells go to the spleen and transition into intermediate 1. And then here, the intermediate 1 seeds the tumor or transitions in case of high antigen into a KLR exhausted phenotype, which is no longer capable to home into the tumor. What we cannot decipher quite yet is if those intermediate 1 cells can give rise to a stem-like population in the lymph node again. That one is still unclear. So with that, I would like to thank the people that did the work. This was really spearheaded by Brendan Horton, a senior postdoc in the lab, and Maria Zaruglia, a recently graduated student. And we have a couple people, Vidit and Teresa, that pick up on this. I would like to thank our collaborators. I would also like to thank our funding, and I'm doing a pun. We're looking for a postdoc, so if there's any senior grad student wanting to move to the East Coast, email me. And I'm leaving you with a cancer immunity cycle, and I'm happy to take questions.
I have a microphone for anyone who wants to ask a question.
Hey, wonderful talk. So what I'm really curious about is sort of, do you see the same thing with vaccination, or would you hypothesize the same thing to be happening with vaccination in your mouse models, just because mucosal vaccinations are becoming a new it thing? And then sort of similarly, if you were to add antivirals or antibacterials, would you then recover some of these immune responses by restoring homeostasis to that mediastinal lymph node? And then lastly, what would you expect to see in humans that are much more dirty at all their lymph node sites?
Unpacking the 3 questions. So to the antiviral, antibacterial treatments, that's on our to-do list. We want to understand, and I want to move away from the germ-free because they're so artificial. That was a proof of concept to see, can we use antibiotic or antiviral treatment to really pin down, is it the presence of the microbiota or is it a certain subset of the microbiota? That's on the to-do list. We need more force. The third question, what would we observe in humans because they're more dirty? Good question. I think our thus spare but existing clinical data suggests it is a phenotype that exists. It's something I think once we have either diversity or specific phyli identified, where we can go back to humans and ask, if you have XYZ bacteria, do you have a higher amount of interferon gamma? Do you have more activated Tregs at baseline? I think it's a cool question. What was your first question?
Vaccine.
Oh yeah, we have actually a grad student who is looking at vaccine administration and how that affects CD8 T cell responses, because we know a lot about vaccines and B cells. We don't know that much about vaccines and T cells, strikingly.
A really nice talk. I had two questions. One following up from what Nick was asking. If you look at melanoma models and metastasize to the lung, do you see a similar phenomena there, just to get an idea of, okay, if you have T cells come to a different site and migrate, do Tregs suppress there? And
there's quite complicated dynamics at play here. So the short answer is, if we put just B16 as a decoy model system into the lung, we see a weaker response than if we put it into the flank. It's still stronger than a lung tumor. Whether there is some antigen-specific overlap between lung antigens and lung cancer antigens, we will clone those Treg TCRs and find that out on the to-do list as well. What makes this even more complicated is if we put the lung tumor in the flank first and then re-challenge for the lung tumor, the flank tumor overrides. But interestingly, when we put the lung tumor in first and then challenge for the flank tumor, the lung tumor dominates. So the site of primary tumor growth really matters, and it's almost like it's similar to the antigen sin hypothesis, that wherever your T cells see the antigen first, they're going into one trajectory versus another. And how we can therapeutically shift that balance back is hard.
Great, thanks. One quick question regarding the second part. Do you have any idea what are the chemokines or the cues that drive the progenitor T cells to go to the spleen from the tumor-draining lymph nodes?
We have no idea. What we know is, because we were really worried this is just a circulatory phenotype, we did very careful time courses to give CD45 IV to make sure it's not circulating T cells in the spleen. They're actually located in the white pulp of the spleen. So there seems to be something of the spleen microenvironment that fosters this interaction. I also don't know how long-lived this intermediate phenotype is.
Thanks.
Thank you. I'm curious what you think about the priming of the Treg, tumor-specific Tregs in the two different models. Specifically, what cells do you think are responsible for priming them? And do you know if there are any phenotypic differences between the migratory DCs that are coming from the lung versus the skin that could, contribute to your model?
So CCR7 is similar. PDL1 is similar on both DC populations. Again, when we take them out of context, both DCs become quite stimulatory. So I think it's a continuous interaction with regulatory T cells. If I would have to guess, I would say the DC1s also prime the Tregs, but that's a guess. We haven't proven this. It's based on the TCR clonotypes that we observe, and some being public between both lymph nodes. I would rule out antigen specificity as the main driver, though this is correlation. We have selected 6 TCRs where we have alpha beta pairs from both locations that we will clone and first do adoptive transfer of effector CD4s with overexpression to see, do we get lung pathology? Do we see longevity? Do they become peripherally induced Tregs? I would have to guess at this point. Happy to report back in a year. Katie.
Hi, Stephanie. I was thinking about the interferon gamma in the lymph nodes, and you said you thought it was innate. I was going to ask if you looked in RAG knockouts, although I realized lymph nodes would probably be very tiny.
Yeah, I don't think we would find lymph nodes. I think we would have to look at TCR beta knockouts, I think, to get at least the B cell component
Yeah.
to be present. No, we have not. We have not looked at any other system. My approach there, because the limiting factor for us to find a cell type that produces the interferon was the intracellular cytokine stain. So we need to go with an actual reporter and because the population will be small.
Then one more question at the back, and maybe this will be our last question as well.
Hello, good morning. Thank you for this opportunity. It was a very beautiful talk.
Thank you.
I'm just brainstormed with this kind of new direction for me. Recently joined postdoc. What led you to hypothesize towards the spleen, because you were interested in the lungs and lymph node and then the spleen, and not the thymus or the other organ? Because how, and the signaling related to this, how could like, you saw the subpopulation and everything, but the signaling that leads to when a tumor bearing environment is having, it's a total different scenario.
It was, I think it was serendipity that we, in multiple assays, observed that the spleen and the lung setting had fewer of those T cells. And it was, I don't know, scientific curiosity to study this further. And my advice would be to keep an open eye for data that you don't expect, because often that's the right path to follow.
Thank you. Thank you.
All right, let's thank Dr. Spranger
Thank
for
you.
a great talk. Thank you.